Hi, it's Miss Parrott, and this video is the first in a series called AP Bio Equations Explained. In this, we're going to demystify the math that you can find on the AP Biology exam. Quantitative reasoning is a powerful tool in the life sciences. We're going to look at why and how we're going to use it in this series. In it, we're going to break down every essential equation and statistical test. We're going to look at how to use formulas step by step with real biological examples, tying the math back to core biology all along the way. It's really about understanding what these numbers mean for the real world and how they can help us determine if our experimental results are significant or not. So, why do I need math and biology? Well, in biology, there's a lot of measuring and observing, so we can use it to help us do things like quantify bacteria, which are too small to see, or determine rates of photosynthesis or of growth. We can use math to model and predict what will happen in the future, like how a disease will spread or how ecosystems may respond to climate change. We can interpret experimental results, again, determining if our results, the differences between our experimental and control groups are significant or not, and correlate data to each other. Also, we can move from qualitative observations, like in this osmosis lab, in these cups with different colored solutions, the carrots grew in mass by a different amount. But we can turn things like big, bigger, biggest into graphs with what's the statistically, like what's the error of their difference, and compare them and make graphs and be able to visualize how they're actually different, and do things like determine the molarity of solutions just by turning big, bigger, and biggest into numbers. Today's focus is on setting the stage. Again, we're looking at why quantitative reasoning is essential in biology. We're going to look at the AP formula sheet and look at different types of equations that involve calculations. Here on the left is the first page of the equation and formula sheet, and on the right is an FRQ that involves a lot of the skills that you need to use the formula sheet for. So first, let's look at the formula sheet. This can be your best friend for quantitative questions if you've used it before and practiced with it. It's divided in sections based on topic. So you can see here at the top, we have statistical analysis and probability. Here are the equations, and then here are the variables that go with those equations. Then there's a chi-square table that'll go with this chi-square equation there at the top. Laws of probability will have to do with genetics. Hardy-Weinberg equation will be looking at genetics and change in allele frequency, so genetics and evolution together. And then there's a section on the bottom right for metric prefixes. At the bottom, it defines mean, median, mode, and range. On the second page, we have equations for rate and growth, which you'll especially need for population dynamics. Again, it defines all the variables. Simpson's diversity index, which will help you understand biodiversity. On the right, this whole column with water potential and solute potential is part of osmosis. And then down at the bottom is surface area and volume, which will help us look at surface area to volume ratio when we're looking at cell size and the efficiency of diffusion. So you'll note there's surface area and volume for a sphere, a rectangular solid, a cylinder, and a cube. I've really only ever seen a cube, but they give you all the equations just in case. The goal of the formula sheet is not to confuse you. It's to help you out. So you don't need to memorize any of this. You do need to be familiar with how to use it and when to apply them. So there's three major types of questions. The first is direct calculation, where you're just going to plug in values from a formula. So in this example, it gives you some flies in a choice chamber, and like where they go after at the beginning and where they are after 10 minutes. And then the question just says, perform a chi-square test on the data for the 10-minute time point in the banana experiment. So you would look at your chi-square equation. You're going to need observed numbers and expected numbers, and these are your observed numbers at the 10-minute mark. And you're expected, and then you just plug in the numbers that you need. The second type is data analysis, where you're going to extract information from sets of data. So you have to pick the right numbers, choose a formula, and interpret these results. So in this example, this table shows the rate of oxygen consumption and ATP synthesis at different temperatures. So you have different temperatures and different outputs, ATP or oxygen consumption. And then the question asks, based on the data in table 1, calculate the average amount of oxygen consumed. So now we know, okay, top row, oxygen consumption, in nanomoles for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees C. So at 25 degrees C tells us we need to use this middle number here, and then we'll do some calculations based on that. So picking the right number and then doing calculations from there. The third type are more conceptual. So let's say we had just done a chi-square test. This question asks us to explain whether our hypothesis is supported by the chi-square test and to justify our explanation. So on that, we would need to know, okay, well, yeah, I have this chi-square value. Now I need to know how to use the chi-square table. Based on my number of groups, what are my degrees of freedom, and which value should I choose? Then, after choosing the right value, you will be able to justify whether your results are statistically significant or not. A quick look at the metric prefixes. So this is one of those things where in the previous question, yeah, this one, right? It gives us volumes in in nanomoles or amounts in nanomoles. Like, what's a nanomole? It's just a mole, and then this can show us that nano is times 10 to the negative 9. So it's super, super tiny. All right, it's a very small amount. Just be familiar with these and pay attention, because you may need to do something simple like convert from centimeters to meters, or something. There may be a unit you don't recognize, like micrograms. This little mu, the symbol mu here shows up. So just pay attention to the units when you're giving your data and when you're giving your answers. If your answers require a unit, you need to have it, because that's a point right there on the exam. Now let's look at the calculators we can use. This has really opened up since I first started teaching AP biology. We used to only be able to use 4 function calculators like this one in the top left. The main thing is that it needs to have a square root button. So now you can use a 4 function calculator, a scientific calculator, or a graphing calculator. But you cannot use a phone or any kind of smart device, anything that connects to Wi-Fi or to the internet, and nothing that has stylus capabilities on it. You can't use anything like that. My main thing with calculators is I encourage you to use whatever calculator you're going to use in the test, use that all year so that you're not hunting for the square root button or the square button. Now, here's the one thing. If you have a 4 function calculator, there is no squared button. So you do have to remember, and there's no formula for it on the equation sheet, that's to get a number squared, you just multiply it by itself. So if you have to do 3 squared on the 4 function calculator, you would need to put in 3 times 3. It's simple, but it tricks kids up if they haven't practiced. All right, for our conclusion, just remember, math, it's not just essential, but it can be a really fun part of biology. The formula sheet is your friend and is not meant to be scary or intimidating. You just got to learn to use it. Be prepared for various question types and get comfortable with your calculator and the different metric prefixes. In the next video, we're going to be looking at mean, median, mode, and range, and how to use these equations to help us answer questions. Thanks so much for watching. Please like, subscribe, and leave a comment or a question about the AP Bio or the AP Bio exam in the comment section below.